Okay, let's pick up where we left off. Here we go. So we are going to talk a little bit about the two major clades of angiosperms and some differences in how the tissues and organs are put together in these different groups. I actually want to make this figure a little bigger here. Okay, so the monocots versus the dicots differ in a few main characters. So we have, first and foremost, one cotyledon in the monocots, that's where they get their name, versus two cotyledons in the eudicots. So, reminder, those cotyledons are the embryonic seed leaves. We also see differences in the arrangement of the vascular tissue in both the stems and the roots. So, oftentimes, vascular tissue will be arranged in what we call a vascular bundle, which is essentially a bundle. No, that's not awful. That's awful. Actually, I can show you a better picture. Vascular bundle, right here. So here's a vascular bundle. It is a little bundle, essentially, of xylem. So there's our xylem and phloem. Actually, then we also have supporting tissues, something like a sclerenchyma cell, for example, which kind of holds the bundle together and makes it very strong. So what you think of as a vein in a leaf or a stem is one of these vascular bundles. So here's another picture of a vascular bundle. So we've got a patch of phloem, we've got a patch of xylem. That's a vascular bundle. So back to our differences here. In a monocot, these vascular bundles tend to be scattered in the stem. Like if we looked at a stem in cross-section, they would be scattered. And in a eudicot, they tend to be in a ring. So that's the stem. Here are the roots. So if we're looking at the roots, the monocot roots have their xylem and phloem in a ring, kind of in alternating patches. And then we've got some pith in here, right? That's that ground tissue that is to the interior of vascular tissue. If we're looking at a eudicot, what we have are patches of phloem, which is blue, shown in blue, alternating in between xylem, what we call arms, because they're just sort of radiating out like this. And we don't have any pith here. We will be looking at examples of these in lab as well. We also have differences in leaf venation, the root system. So we have what we call a fibrous root system in monocots and a taproot system in eudicots. Differences in floral organs, flower parts in threes or multiple thereof in monocots, flower parts in fours or fives or multiples thereof in eudicots, and differences in number of pollen grain apertures, 1 in monocots, 3 in eudicots. So quite a few differences between these groups. So let's talk a little bit about plant growth, because it is different than the way an animal grows. So plants exhibit something called indeterminate growth. Which basically means they keep growing throughout their lifetime. Right, think of something like a tree as compared to something like a human. Right, that tree, season after season, keeps elongating stems and branches and roots and offshoots of its roots. Animals don't do this, right? The reason that plants have this indeterminate growth is because they have perpetually undifferentiated cells known as meristems. So a meristem is an area of undifferentiated cells that are capable of dividing to produce new cells that can mature into any cell type. So there are two major kinds of meristems in plants. There's what we call the apical meristems. We would find these at the tips of roots and the tips of shoots. They are responsible for growth in length, or what we would call primary growth. Right? So they cause shoots to grow up and roots to grow down. There are also meristems known as lateral meristems. Lateral meristems are responsible for growth in girth, so growth outward, also known as secondary growth. So let's take a look at this. So up at the tip of this plant here, we have what is called the shoot apical meristem right here, where we have lots of undifferentiated cells that are capable of dividing to produce new shoot tissues. Also at the tips of roots, we have what's called the root apical meristem, which is the same thing, except for it's responsible for the primary growth in the roots. Now, not all plants are capable of secondary growth. Not all of them have it, but those that do have lateral meristems that will allow growth in girth, the thickening of stems or roots. So let's zoom in a little bit and look at a root and look at how growth in a root happens. First, talking about the primary growth in roots, growth downward. So the tip of the root is covered by what we call the root cap. So this is a protective layer that protects the apical meristem from damage. So it keeps the apical meristem from getting damaged. Because if the apical meristem gets damaged, that's really a problem because it means that the cells it's making are going to be messed up. The root cap secretes a slimy, lubricating polysaccharide that helps the root move through the soil a little more easily. So the very tip of the root is not the apical meristem. It is this area in pink here called the root cap. So after we get past the root cap, we have this root apical meristem in red. So that's our root apical meristem. So growth in the root occurs in 3 zones. There's what we call the zone of cell division, starting just with the apical meristem and making it about here. There's the zone of elongation and the zone of maturation. And you'll notice they overlap with each other. So the zone of cell division is where new daughter cells are being produced and start to differentiate. That is the zone of cell division. The zone of cell elongation is where these newly made cells are going to elongate. They're going to basically take up a bunch of water and start to become much, much larger. And this elongation is one of the things that really helps push the root down into the ground as this area elongates. And then, not surprisingly, the zone of cell maturation are where these cells are going to fully differentiate and mature into whatever cell type they're going to be, whether that be ground tissue, epidermal tissue, or vascular tissue. And in this zone of cell maturation, we start to see the root hairs, right? Because you need to be a mature cell before you can differentiate into a root hair. Remember I said I'd show you root hairs? Here we go, I kept my promise. A root hair, again, an extension of a dermal cell in order to create more surface area over which to harvest water. So most gymnosperms, let me back up a second here, and eudicots have what's called a taproot system, and these taproots will develop what are called lateral roots, and that is what you can see coming off the side here. We actually can get a lateral root or a side root forming. Now I wanted to take a second and compare a monocot root to a eudicot root. I mentioned this a little bit already, but let's take a closer look. So in our monocot root, we have our xylem and phloem in kind of these alternating patches. So we have patches of phloem up here, and we have this alternating with xylem, which is usually much bigger, by the way. And in the middle, we have our pith. Now, a term that I haven't really talked about yet, we like to call this whole collection of vascular tissue the stele. So that's just the xylem and phloem collectively. So the stele in a monocot root looks different than the stele in a eudicot root. In the eudicot root, we have what we call the xylem arms, kind of these, in this case, looking very much like a cross shape, and then we have the phloem in patches in between those arms. So there's our phloem here. So the stele looks quite different in a monocot versus eudicot root. So here's a few more images. So here is our eudicot root with its typical taproot structure, and here's our monocot root with its fibrous root structure. And here's a newly germinating root, and you can see those really super fine root hairs on it. So we also get really neat modifications of roots for different types of adaptations. So we have what are called storage roots. So think of something like a carrot or a radish, where the root has actually become like a little more fleshy and contains more sugar or starch and is edible. That would be a storage root. We have roots that can help take in more oxygen. So pneumatophores are common in plants that are growing in very low oxygen soils. And so they actually come up above the soil or water surface so that they can absorb a little more oxygen. We have roots that are for support, so prop roots, which we see in this plant. Also, these are common in corn plants. So anytime you have like a really long skinny stem like this, you risk the plant falling over in the wind. So sometimes you get these prop roots that come up really high out of the soil to help give it a little more stability. Buttress roots are another example of a support root. So trees that live in a really shallow soil might develop these really thick, broad buttress roots that give the plant more stability. Finally, we also have aerial roots that will develop sometimes from the branches of trees and grow downwards. And so this is a strangler fig. And so sometimes they actually can wrap around other plants, or in this case it's wrapped around a structure, and will grow above the ground. Okay, so we talked about primary growth in roots. Let's talk about primary growth in shoots. So the shoot apical meristem is this dome-shaped, oops, that's my eraser, not my pen, is this little, stop it, this little dome-shaped mass of cells at the tip of a shoot. It gives, on either side, we have leaf primordia that give rise to leaves. We also have in our shoot these little axillary bud meristems that are going to give rise to side branches. So each of these little shoot apical, or each of these little axillary bud meristems have their own little teeny tiny dome-shaped shoot apical meristem and are capable of producing a side branch. Now, sometimes they don't, and this is because of something called apical dominance. So sometimes these will branch off and sometimes they won't, depending on how much apical dominance is going on. So apical dominance, basically the hormones produced by this shoot apical meristem can inhibit these axillary bud meristems. And this is why sometimes if you're growing a plant like basil, if you want it to get bushier and have more side branches, pinch off the tips. Pinch off this shoot apical meristem. That cuts off the hormonal signal that is keeping those axillary buds meristems from happening. So that's why pruning can make something bushier. You're removing the shoot apical meristems. All right, so let's talk a little bit about the arrangement of vascular tissue in stems, in shoots. Because again, monocots versus eudicots, we have some different arrangement going on here. So in monocots, we have those vascular bundles of xylem, which is usually bigger, and then our smaller phloem. This is typically going to be surrounded by some really strong sclerenchyma cells or something else that helps kind of strengthen this bundle. These are going to be scattered in a monocot stem, and in a eudicot stem, they're going to be arranged in a ring. So just kind of an important difference between the two. And this will be significant later because, as I'll get to a little bit later, eudicots are capable of secondary growth in their stems and monocots are not. So this ring-shaped pattern of the vascular bundles is one of the things that's going to allow the eudicots to put on secondary growth. Okay. So I just wanted to throw another couple terms out here that are kind of important, and I apologize that my figure is kind of running together here. So I mentioned we have this apical bud at the tip here, and that we also have these axillary buds, right, that are technically capable of producing an offshoot. So an area where we have these intersections, where we have this bud, is known as a node. So here is a node, here is a node, here is a node, here is a node. And the area in between nodes is known as internodes. These terms are going to come up a little bit in lab. So like roots, we sometimes get some interesting modifications of our stems. So we can have stems that are modified to be underground, and when that happens, we call it a rhizome. So a rhizome is an underground lateral stem, or a horizontal stem. Oops. Oftentimes, not always, but oftentimes, it's also kind of fleshy and can store things. We also find in, for example, onions, a super highly compressed stem. So that is the stem of an onion. Super duper highly compressed, and then actually most of what the onion is are storage leaves. So leaves can also be modified. And then we also have what are called stolons, which is another kind of horizontal stem, this time above ground. And think of a strawberry. Puts out these really long stolons, and at the end will typically have a little baby plant. And this is one way that this strawberry plant can kind of invade different areas of the habitat. And And we also have some stems that are straight up for storage. So something like a tuber of a potato. A potato is a modified stem for storage. All right, we're going to take a little break here, and we're going to come back and talk a little bit about leaves.